So welcome back, and the topic of discussion that we are going to have is we'll be talking about small intestine, right? The histology of small intestine. Now before getting into the topic, let us see what are the structures in small intestine. The structures in small intestine are our duodenum, jejunum and ileum, right? We'll be talking about their similarities and their difference in histology, right? So the thing that we'll go toward is we'll go through all these things, right? We'll go through all these things. So now let us begin our discussion. As we said, the three parts of small intestine: duodenum, jejunum and ileum, right? Now let us talk about their similarities. Talking about their similarities, the thing is that these structures all, right, the structures present in small intestine all are designed to increase the absorption, right? Increase the absorption. Okay, now in the process of increasing absorption, okay, you can see right here, this is our diagram. What is this? This is all our duodenum, right? This is our duodenum, this is our jejunum, right, and this is our ileum. Right now we are talking about similarities. Talking about similarities, all these three structures, right, every structure in small intestine, they consist of what? They consist of what? They consist of same layers of what? Same layers. They consist of same layers. Now what are the layers that we need to talk about? The layers are, right, the innermost one. What is our innermost one structure? The innermost structure is our mucosa, right? The innermost structure is our mucosa. Similarly, outer to mucosa we have submucosa, outer to submucosa we have muscularis externa, and in this muscularis externa you can see there are the outer, right, outer are outer are longitudinal, whereas the inner are circular. Okay, now moving further, we have the outermost one, and the outermost one is our what? The outermost is our serosa, right? Okay, now what I said is, talking about similarities, what we have is we have the innermost one mucosa, right, and the outermost one serosa, right? Similarly, inner to mucosa we have submucosa, and then we have muscularis externa, right? Okay, now these are similarities. And talking about another, yes, I have already mentioned it, right? Talking about other thing, that these structures are all designed to increase the absorption, right? Increase the absorption. Now okay, now if you see the pattern, right, if you see the pattern, these absorptive area, right, talking about this absorptive area, what are the absorptive area? You can see the innermost one mucosa, right? This is mucosa. As you can see, these in that mucosa we can see there are many circular folds. You can see many circular folds, right? Now okay, now there comes the term Pelican Circularities, right? Pelican Circularities. What are these Pelican Circularities? These are mucosal fold. Yes, we talked about mucosa right here, the innermost structure, right? These are muscular fold. And why are these folded? I mean, why? What is the importance of muscular fold? That the mucosal fold, it's important is that it help in increasing of surface area, right? Surface area. Okay, now another thing that we need to know here is there are villi and microvilli, right? Villi and microvilli. Now what are these villi and microvilli? You can see these, right? You can see these are villi and microvilli, right? Villi are these structure, and under these structure there is constricted patches, right? I'll show you in this diagram, right? These structures. What are these structures? You can see villi more carefully in this diagram, right? Okay, villi. What are villi? You can see finger like projections, you can see right here. These are what? These are our villi, right? And these are our villi. Now what about microvilli? You can see here, right? Now what are these? These are striated border. Microvilli are striated border. So these are villi and microvilli, and these are present all over in our what? Small intestine, right? Small intestine. Okay, now talking particularly, right, we said that these are similar structures, right? Similar structures. Now yeah, we're talking about Pelican Circularities, right? These are what? Mucosal fold increasing in surface area, right? And we talked about villi and microvilli, right? Now talking specifically about what? Pelican Circularities. Now what are these Pelican Circularities doing? These are also same, right? What are these? These are our what? Circular mucosal folding, right? Circular mucosal folding of mucosa, and these have same function. They also, we talked under what? Increasing in surface, increasing in what? Increasing in surface area, increasing in surface area. So if you look toward Pelican Circularities, right, these all structures, right? These all structure Pelican Circularities. You'll find that the Pelican Circularities starts developing at the place of duodenum, right? I mean, they will start at duodenum, right? They are they start developing in duodenum, and then they become maximum in the case of jejunum, right? They become so maximum, and they will get reduced in the case of villi. So we can say that, sorry, they will get reduced in the minimum in the case of ilium, right? What I said is, it becomes development at duodenum, right? It will be at maximum in jejunum, and it will be least at ilium. So we can say that they will be reduced, right? They will be reduced slowly and gradually as we descend downward. Okay, we talked about this thing. Now, yes, we already mentioned about the common features, that these have similar layers, and the layers are mucosa, submucosa, muscularis externa, and serosa. So these were the things that we're talking. Now, moving forward in this diagram, in the same diagram, what we can see is, let us talk about duodenum. As you can see, right, in this duodenum, right, in this duodenum, what is this? Our innermost layer, right? This is our mucosa, right? In mucosa, and below that mucosa we have submucosa. You can see right here, submucosa. In this layer of submucosa, you can see what? You can see duodenal submucosal gland, right? Duodenal submucosal gland. Now, what is this exact position in which this duodenal submucosal gland are found? Talking about these exact location, these duodenal submucosal gland, these are present in proximal duodenum. Where? Proximal duodenum, right? Proximal duodenum. Now, what are these duodenal submucosal gland exactly called? These are called Brunner's gland. Brunner's gland. Okay, we'll go in detail sometime later. Okay, now these Brunner's gland are the exclusive feature. These are what? These are the exclusive feature of our duodenum. So, talking about the histology of duodenum, this is the most important thing that we need to mention in any case. Okay, now moving a bit, moving toward jejunum. As I've already said, jejunum consists of what? Highest amount of Pelican Circularities, right? Okay, Pelican Circularities. Now let us move toward ilium, right? Talking about ilium, what we have to say here is, in ilium, the exclusive feature of ilium is our what? These Pierce patches. Now, where are these Pierce patches present? These Pierce patches are present, right? Okay, these Pierce patches, what are these Pierce patches? These Pierce patches are what? Aggregated lymphoid nodules. Aggregated lymphoid nodules. And talking about these Pierce patches, what are these Pierce patches? These are MALT. M-A-L-T. Now, what are these MALT? These are mucosa associated lymphoid nodules, right? Mucosa associated lymphoid nodules, or mucosa. The word mucosa, that means these are present near to mucosa associated lymphoid nodules. Now, Pierce patches are what? Associated or gathering of what? Our lymphoid nodule, right? Pierce patches. Okay, and we'll talk about these things also in detail afterwards. Now, okay, now the thing that we need to keep know in this diagram is, okay, now we need to know the exact place where these Pierce patches are found. And talking about the exact place where these Pierce patches are found is, they are found where? They are found toward terminal ilium. Terminal ilium. Okay, we mentioned about what? We mentioned about Brunner's gland. Where were Brunner's gland found? Brunner's gland were found in proximal duodenum. And talking about what? Talking about our these Pierce patches. Where are these Pierce patches found? They are found in the case of terminal ilium, right? One at the terminal ilium, but Brunner's gland at the proximal duodenum. Okay, now in this diagram, we are clear, right? We are clear. We had a brief idea about what is going on. Okay, now let us move forward. Yes, moving forward, we have this diagram. Now, what is this diagram? This diagram is our what? The histology of what? Duodenum. Okay, duodenum. Now, in this diagram, the thing that we need to look here is, we need to look about, yes, we talked about what? Villi, right? What are these villi? These villi are what? These villi are our mucosal folds, right? Mucosal folds, right? And they are designed to increase surface area. Okay, now as we mentioned, right, these structure, what are these? These are what? Striated borders, and these are striated borders, and they are striated to form what? Microvilli. Microvilli. Okay, now moving further, as we come a little bit down, we will see intestinal crypts. Now what are these intestinal crypts? These, right, these are the structure, right, they are present between the villi, or we can say these are present between the villi, and these are responsible for to form intestinal glands. Intestinal glands. Okay, right, these are forming intestinal glands. Now we were still till now we were talking within the topic of mucosa. Now moving towards submucosa, yes, we'll find duodenal glands. Now these duodenal glands are our Brunner's gland, as we have already talked, Brunner's gland, right? And below the submucosa we have we have Muscularis externa, and then the last layer we have is serosa. So now let us talk about specifically about the cells, right, about the cells. Now talking about, okay, as I've said, what I've said is these intestinal polyps, right, these intestinal crypts, right, these intestinal crypts, what they are doing is they are responsible for forming what? Intestinal glands. Now let us talk about intestinal glands, right? Okay, let us talk about the histology of intestinal gland. Okay, here the first thing that we need to look right here is we need to look about enterocytes. Now what are enterocytes, right? These are cells, right? These are cells, right? The cells in our intestine. Now here each these cells longitudinal, right, long cells, or these are our columnar cells. These columnar cells are enterocytes. What are these? Enterocytes. These are the long columnar cells, right? Okay, now what I'm saying is what I said till now is these intestinal gland. What are these intestinal gland? These are the structures, right, that will be formed within our crypts of Liverquin, right? Okay, now these we talked about enterocytes, right? Enterocytes. Now these columnar cells or enterocytes, what is these role? These play a role in absorption, right? They will play a role in absorption. Now moving further, what we need to talk about is we need to talk about our goblet cells. Now what are these goblet cells, right? Okay, right now we are talking about intestinal gland, right? Under that intestinal gland we are talking about goblet cells. Now what are these goblet cells? These goblet cells are responsible for forming mucus, right? Mucus. Okay, you can see in these goblet cells, what you can see is, you can see here, right? You can see, okay, I'm showing right here. You can see foamy structure, right? You can see foamy structure. Now why are these foamy, right? Why they are foamy in appearance? They are foamy in appearance because of mucus. Right? Because of mucus. Okay, now, okay, and you can see goblet cell right here. You can also see right here. These are foamy structured, right? You can see right here. Okay, these are foamy, right? Foamy because of mucus deposition. Now let us move to our third number cells. Now third number cells is our enterocrine cells. Now what are these enterocrine cells? Enterocrine cells are responsible to give hormones, right? Hormones. Entero within intestine, and endocrine means endocrine. What does endocrine system does? It will produce hormone. Now moving further, what we have is we have Paneth cells. Now these are important, right? What are these? These are our Paneth cells. Now as you will see Paneth cells, you will find pink. You can, okay, this is our one Paneth cells. This is our another Paneth cell, and this is our another Paneth cell. You can see, right? You can see right here Paneth cell, right here Paneth cell. Now how to identify these Paneth cells? Now in it talk about that. Now, the thing that we talk right here is, okay, what we talk till now, we talk about what we are on the intestinal gland. On the intestinal gland, we talk about what we talk about enterocytes, we talk about goblet cells, endocrine cells, cells, right? Now let us go towards this. Talking about a description, what we talk about, what are the enterocytes? These are columnar cells, right? They help in absorption. Okay. What about goblet cells? Secretes mucus, right? And talking about family cells, what these family cells are? These are what? These are responsible for providing immunity, right? How they are providing immunity? Because they are giving access of pathogens or amoeba bacteria. And what they do is they secrete what? Cytokines. And what these cytokines contains? These cytokines contains human necrotic factor also. They are responsible for creating these access points. Now, by giving access of these pathogens, what they are doing are they are maintaining normal intestinal flora, right? Okay. Why they were epithelially dysmorphia, as you can see, they were epithelially dysmorphia because they were containing what large amount of aggregation of lysozymes, right, on the edges. That's what I said. These lysozymes, what they are doing, they are producing cytokines, and these cytokines are what responsible for creating immunocoric factor. That's all. Moving toward another cell, M cell, that we did not mention right here is, okay, we did not mention right here about M cell. Now what are these M cells? These M cells are what? These are microvilli cells. Now what is this important? It's important that it contains what? Antigen. These are antigen presenting cells. They are presenting what? They are presenting what? They are presenting our what? Our DSP. They are what they are doing? They are presenting antigen to our cells. Okay. Okay. Right? Okay. Now moving further, what we have is we have NPC. Now what is these NPC? These are neuroendocrine cells. What are these? Neuroendocrine cells, right? Neuroendocrine cells. Okay. Now these NPC cells are not found in our intestine or small intestine. These are neuroendocrine cells and these are present also in our kidney, right? Now what is this NPC? These are these neuroendocrine endocrine cells. They are responsible for producing what? Hormones, right? And what about skin cells? protective and protective, right? And it helps in treating of uterine ulcer in which there was destruction or destruction due to the mucosa, right? Mucosa. Now again, let us refer to whatever we talk about villi. What are these? These are surface protection of mucosa, right? Now between the villi we talk about crypt of ureters, right? Or intestinal crypts. Now what we say in that intestinal crypt is the noun glands. Intestinal glands are seen in our what? Intestinal crypt of the subdural glands are seen here. And there are also stem cells. What is stem cells? Simply we say proliferating them. And what they prepare is they prepare the cell of intestine, and the cell of intestine is nothing but the intestine, right? It also refers to our goblet cells. Now talking about enterocytes, now what are these? Enterocytes is simple for the epithelium like villi, and it is important for insides. Talking about goblet cells, what are these goblet cells? These goblet cells, right? Okay, there are two cells, right? I mean to just make it clear, one is goblet cells and another is Paneth cells, right? Paneth cells. Now talking about goblet cells, what is happening is these goblet cells migrate from base, right? From base to apex. See the position of goblet cells toward apex. What indicates that cell they are facing toward the base, right? They are migrating from apex to the base. Okay. What is it? What are the location of goblet cells? They migrate from base toward apex, right? And talking about Paneth cells, as I have already mentioned, what are these Paneth cells? These migrate from apex toward the base. Yes, we have seen everything right here, right? Paneth cells they were sitting toward the base, goblet cells they were sitting toward the apex region, right? Okay. Now, right here, the thing that we are talking that here is we're talking about Paneth cells, right? Okay, as I have already mentioned, goblet cells, why they are forming in structure, why they are forming in structure, because they consist of vessels, right? Okay, now migrate from this Paneth cell migrate from apex toward the base. As you can see at the base we have Paneth cells. Here it is not colored. If it was colored, what you will see is you will see apical region over here, right here. Apical region. Why these are apical in why they are distribution of apical region? Because they contain lysozyme. They are what phagocytic in nature because they secrete cytokines. Cytokines which are rich in what? Tumor necrotic factor. Now this tumor necrotic factor, all what they will do is they will kill excess of microorganism, and excess of microorganism will be killed and our flora will be maintained. Okay, we talked about M cells. What are these M cells? These are antigen presenting our what? To our lymphocytes. Similarly, we talked about duodenum. We talked about what? In submucosa there is one exclusive donor gland, right? And these are exclusively present in our proximal part of duodenum, right? Okay, now okay, there. Okay, content of the stomach is highly acidic, yes, because of HCl, and additional mucosa is required to neutralize it, and that additional mucosa is provided by our Brunner's gland, right? These Brunner's gland are described what is anti-parietal function. And duodenum, there is no Brunner's gland, and duodenum contains no Pierce patches. Yes, we talked about Pierce patches in the case of Ilium, right? Ilium. Okay. Now, we talked about Goblet cells present in both small and large intestines. That means goblet cells are most like parasites and body matter. As we descend downward in our intestine, the goblet cells are more prominent. You can say like body matter secretions from this. This was all about what it was all about.